Hi everybody, and welcome to our module on the electron transport chain. The electron transport chain is an ATP generating process that occurs inside the mitochondria of our cells. It takes molecules like NADH and FADH2, which are electron carriers, and it takes their electrons and passes them through a series of structures in order to generate ATP for energy for the cell. Before we get into electron transport, let's pause and talk about aerobic metabolism. This is the process by which cells use oxygen to convert glucose into the maximum amount of ATP. Recall that glycolysis converts glucose into two molecules of pyruvate, and in doing so it generates two ATPs and two molecules of NADH. The pyruvate can then leave the cytosol, which is where glycolysis takes place, and enter the mitochondria. Inside the mitochondria, pyruvate can be converted to acetyl-CoA. Acetyl-CoA can then enter the TCA cycle and be converted to GTP and also NADH and FADH2. And it is these molecules of NADH and FADH2 which can be used by the electron transport chain to generate ATP. Now in this module we will talk about how electron transport uses NADH and FADH2 to generate ATP, but first we have to talk about something else, which is how these two NADHs right here generated by glycolysis get into the mitochondria so that they can join these other molecules here to participate in the electron transport chain. The problem that NADH has is that it cannot directly cross the mitochondrial membrane to get inside the mitochondria and participate in the electron transport chain. So to work around this problem, cells have something called the malate shuttle. So the malate shuttle gets its name because the first step is conversion of oxaloacetate into malate. In doing this, NADH is converted to NAD+, and what this essentially means is that NADH transfers its electrons to oxaloacetate, and that creates a molecule of malate. Malate can cross the mitochondrial membrane through a shuttle, and once it gets inside, the malate is converted back to oxaloacetate, and that generates a molecule of NADH. So what we have essentially done here through the malate shuttle is move NADH from outside of the mitochondrial membrane to inside the mitochondrial membrane. Now the oxaloacetate that's inside the mitochondria is then converted into an amino acid called aspartate. In doing so, it creates a molecule of alpha-ketoglutarate, and then aspartate can cross back into the cytosol via a transporter. Alpha-ketoglutarate can also cross back into the cytosol, and once back in the cytosol, the aspartate and alpha-ketoglutarate are converted back into oxaloacetate, and the shuttle can be used again. So this entire process basically just takes molecules of NADH, which cannot directly cross the mitochondria, transfers their electrons around, and generates NADH inside the mitochondria, which can then go on to participate in the electron transport chain. The second method by which NADH can transfer its electrons into the mitochondria for use in the electron transport chain is called the glycerol phosphate shuttle. This involves an enzyme called glycerol phosphate dehydrogenase that is present both in the cytosol and also in the mitochondrial membrane. In this case, we start with a molecule of NADH that is present in the cytosol from glycolysis, and we convert it into NAD using the enzyme glycerol phosphate dehydrogenase. This enzyme will convert a molecule of dihydroacetone phosphate into glycerol phosphate. The mitochondrial version of the membrane then will convert this back, and in doing so, it will generate a molecule of FADH2 inside of the mitochondria, which can directly go and participate in the electron transport chain. The difference here between this and the malate shuttle is that the NADH has been converted into a molecule of FADH inside the mitochondria, and FADH cannot generate as many ATPs as NADH can, and thus we've given up some of the energy potential of glucose by using this particular shuttle instead of the malate shuttle. Once all our molecules of NADH and FADH2 are inside the mitochondria, they can then participate in electron transport. This process will extract electrons from the electron carriers NADH and FADH2, and it will transfer those electrons to oxygen. That's why this is called aerobic respiration or aerobic metabolism. In the process of transferring those electrons to oxygen, this process will generate or capture the energy from those electrons and convert it into ATP. NADH can be oxidized to NAD plus a hydrogen ion plus 2 electrons. FADH2 can be oxidized to FAD plus 2 protons plus 2 electrons. And then those 2 electrons from either molecule can be added to 2 hydrogens and an O2 molecule to create water. And that's what we're going to do, is give those electrons to oxygen and create water, and in doing this we will generate ATP. The process of electron transport is performed by four complexes, which are structures embedded in the inner membrane of the mitochondria. Recall that the mitochondria has a double membrane separating it from the cytosol. The cytosol is out here. There is an outer membrane and then an inner membrane, and the two membranes are separated by an area called the inner membrane space. The four complexes of electron transport are called 1, 2, 3, and 4. They're embedded in the inner membrane, and they each play an important role in extracting energy from the electrons in NADH and FADH2. And what I'm going to do in the next few slides is talk about each of these complexes one at a time and what they do in electron transport. So let's start with complex 1. This is also called NADH dehydrogenase. One of the most confusing things about electron transport is that all the elements have more than one name, and you have to know both names. So complex 1, also called NADH dehydrogenase, will oxidize NADH and convert it from NADH into NAD+. In doing so, it will transfer the electrons from NADH to coenzyme Q, which is also called ubiquinone. Remember, everything has two names. So I've drawn a picture of what happens in complex 1 down here at the bottom of the screen. The electrons from NADH are transferred to coenzyme Q by complex 1. Once complex 1 has passed the electrons from NADH to coenzyme Q will shuttle those electrons to complex 3, skipping over complex 2. In the process of doing this, hydrogen ions are pumped into the inner membrane space, and we'll talk about in a few slides how this will be used to generate ATP. Now, complex 1 is made up of multiple different molecules. That's why it's called a complex. Some of the key intermediates involved in transferring electrons from NADH to coenzyme Q via complex 1 include flavin mononucleotide and iron sulfide compounds. Basically, what's happening is the NADH is passing its electrons to a series of substances that are part of complex 1, and then they finally get passed to coenzyme Q, which goes on to complex 3. Here's a picture just to make this clear in your mind. NADH passes its electrons to coenzyme Q, which is found within the inner membrane. Coenzyme Q then transfers those electrons on to complex 3. In the process, hydrogen ions are pumped into the inner membrane space where they will later be used to make ATP. On your clinical rotations, you'll see many patients taking coenzyme Q supplements. So why are they doing this? Well, the reason is because there are some data that indicate that statin drugs used to treat high cholesterol decrease levels of coenzyme Q inside of cells. It's hypothesized that this contributes to the statin myopathy. Many patients on statins experience muscle soreness, and it's thought that the low levels of CoQ may contribute to this. So the CoQ supplements are taken because they may help in theory. You should know, however, there are no good data supporting this. But I wanted to point out to you that you will see patients taking coenzyme Q as a supplement when you do your clinical rotations. Now let's talk about complex 2 of the electron transport chain. Remember that everything has two names, and the other name for complex 2 is succinate dehydrogenase, and that's because it's an enzyme involved in the metabolism of succinate as part of the TCA cycle. This enzyme serves dual purposes. It's part of the electron transport chain as complex 2, and it's also part of the TCA cycle. It transfers electrons from succinate to FADH2, which can then go to coenzyme Q. If you look at this picture on the screen here, this is complex 2 or succinate dehydrogenase. They're both the same thing. It will convert succinate to fumarate, and in doing so, it will generate a molecule of FADH2 and then immediately pass those electrons on to coenzyme Q. And as I told you before, coenzyme Q goes on to deliver those electrons to complex 3. Complex 3 has another name, just like everything else. It is also called cytochrome BC1 complex, and the job of complex 3 is to take those electrons from coenzyme Q and transfer them to another shuttle for electrons called cytochrome C. Just like coenzyme Q shuttles electrons between complexes 1, 2 on the way to 3, cytochrome C is going to shuttle electrons from complex 3 to complex 4. In the process of doing this, it will pump hydrogen ions into the inner membrane space. Here's a picture just to make this very clear in your mind. Complex 3 takes the electrons from coenzyme Q and transfers them to cytochrome C, which is found in the inner membrane space. Cytochrome C then transfers the electrons to complex 4. In the process, hydrogen ions are pumped into the inner membrane space. Let's pause here for a second and talk about what we mean when we say that something is a cytochrome. The cytochromes are a class of proteins that contain heme groups. Heme groups are iron plus a porphyrin ring. This is a picture of a heme structure on the screen here. It has iron in the middle, and this big structure that surrounds it is called a porphyrin ring. In hemoglobin, most of the iron is found in the Fe2+ state. Iron can be either in the 2+ or 3+ state. In cytochromes, however, the iron goes back and forth between the 2+ and the 3+ state. This makes it very favorable for use in electron transport because the oxidation state can change as electrons move through the cytochrome structures. There are many different cytochromes found inside of cells in the body. Cytochromes A, B, and C are used in electron transport, and then you may know that cytochrome P450 is very important as part of drug metabolism. So back to the electron transport chain, now let's talk about complex 4. Just like all the other complexes, it has a couple of different names. It's sometimes called cytochrome A plus A3. It's also sometimes called cytochrome C oxidase because it reacts with oxygen and therefore oxidizes cytochrome C. This complex contains copper, and it combines the electrons that have come at it from the other elements of the electron transport chain with oxygen, and therefore this is the complex that generates water. It also pumps hydrogen ions into the inner membrane space just like some of the other complexes. So here's a picture to try and put the whole process together for you. Complex 1 can transfer electrons to coenzyme Q, which passes them along to complex 3. In addition, complex 2 can also transfer electrons to coenzyme Q. Complex 3 then passes electrons on to cytochrome C, which passes them on to complex 4, and then complex 4 passes the electrons on to oxygen to make water. Throughout this process, hydrogen ions are being pumped into the inner membrane space where they will accumulate and later be used to generate ATP. So now let's talk about how ATP is generated from all those hydrogen ions sitting in the inner membrane space. But before we can do that, we have to define a couple of terms related to the process of phosphorylation. There are two general ways by which ATP is produced. The first way is called substrate level phosphorylation, and the second way is called oxidative phosphorylation. Substrate level phosphorylation occurs via an enzyme. Take, for example, this step in glycolysis, where phosphoenolpyruvate is converted to pyruvate. This conversion adds a phosphate group to ADP to create a molecule of ATP. This is called substrate level phosphorylation. You are phosphorylating or adding a phosphate group to the substrate of ADP directly to create a molecule of ATP. Oxidative phosphorylation, on the other hand, is something that only happens in the mitochondria. It is the process by which all these hydrogen ions, which were pumped into the inner membrane space by electron transport, are used to generate ATP. In oxidative phosphorylation, the enzyme ATP synthase, which is also sometimes called complex 5, will allow these hydrogen ions to leak back inside the mitochondria, and as they come across, they will be used to synthesize ATP from ADP. There's a lot of energy stored inside all these hydrogen ions from this gradient across the membrane, and that energy will be used to drive the synthesis of ATP. Let's talk in a little bit more detail about ATP synthase. As I said before, it's also called complex 5. Remember, everything involved in electron transport and oxidative phosphorylation has more than one name. And it converts the proton gradient, which is really an electrical charge gradient, into ATP. This is called using an electrochemical gradient to drive the synthesis of ATP. It's also sometimes described as a proton motive force. The protons are essentially moving down their concentration gradient. They are concentrated in the inner membrane space, and they are diffusing through ATP synthase into a place where they are less concentrated. This process is sometimes called chemiosmosis. The PO ratio is the amount of ATP generated per molecule of O2 that undergoes oxidative phosphorylation. And classically, it was thought that the PO ratio had to be an integer, and from this they derived that 3 ATPs were generated per NADH and 2 per FADH2, and a lot of textbooks still report these numbers. There are some newer estimates that it may actually be closer to 2.5 per NADH and 1.5 per FADH2, so you will see some textbooks that report these numbers, and that can be confusing. I've listed a review article on the screen here which gives a nice description of how they determine the PO ratios and why they're reported differently. If we go back to our overview slide, we can now determine how many molecules of ATP can be synthesized by a molecule of glucose that goes all the way through aerobic metabolism. You know that we get 2 ATPs per glucose from glycolysis itself. In addition, each molecule of acetyl-CoA that enters the TCA cycle generates a GTP, which can be used to create an ATP. The NADHs and FADHs from the TCA cycle can be used to generate 11 ATP via oxidative phosphorylation, and in getting this number I've used the integer values for PO ratios. And then finally, the two NADHs that come from glycolysis can be converted into two NADHs inside the mitochondria worth 6 ATPs if you use the malate shuttle, or they can be converted into two FADHs via the glycerol 3-phosphate shuttle, and that is worth 4 ATPs. If you sum all these numbers together, you determine that we can get either 30 or 32 ATPs per molecule of glucose, depending on which shuttle we use to get these two NADHs inside the mitochondria. For the remainder of this module, I'll talk about drugs and poisons of oxidative phosphorylation. In general, there are two ways to disrupt oxidative phosphorylation. The first way is to block or inhibit the electron transport chain. If you interrupt the passage of electrons from one complex to another, you will disrupt oxidative phosphorylation. The other way to do it is to allow those hydrogen ions to leak out of the inner membrane space. This is called uncoupling of electron transport, because electron transport still occurs, but it is no longer coupled or linked to oxidative phosphorylation because all those hydrogen ions have leaked out of the inner membrane space and cannot be used by ATP synthase to generate ATP. There are a couple of inhibitors of the electron transport chain you should know about. The first one is called rotenone. It's an insecticide that binds complex 1, and it prevents electron transfer to coenzyme Q. It's described as an inhibitor of the reduction of coenzyme Q. Another inhibitor of electron transport is antimycin A. It's an antibiotic that is not used clinically. It can bind to complex 3, the BC1 complex. Then finally, there are two important substances that can bind to complex 4. Remember that complex 4 transfers electrons to oxygen, and it contains iron in both the 2+ and 3+ state. Carbon monoxide likes to bind to iron in the 2+ state, and remember that's the state that iron's found in hemoglobin. So the main problem from carbon monoxide poisoning is that it binds to iron in hemoglobin and prevents oxygen uptake by red cells. But a lesser effect of carbon monoxide poisoning is that it binds the 2+ iron in cytochrome A3 and competes with oxygen for electron transfer in complex 4. A more important poison of complex 4 is cyanide. Cyanide binds cytochrome A3 in the Fe3+ state, and this makes it a very important disruptor of electron transport. It is not going to have much effect on red blood cells because hemoglobin has mostly Fe2+ iron, and cyanide cannot bind to the Fe2+ iron. So let's talk about cyanide poisoning in a little more detail. Some of you may know that cyanide was used in gas chambers in World War II. It can be deadly if patients are exposed to large quantities. And the reason it is deadly is because it binds to that Fe3+ iron in complex 4 and disrupts electron transport. Patients who are exposed to cyanide get CNS symptoms that are relatively nonspecific, like a headache and confusion. In the cardiovascular system, they get tachycardic and hypertensive, although this can progress to shock. And initially, they will breathe very quickly, but then they can progress to respiratory shock. Classically, the venous blood is bright red. That's because oxygen is not getting extracted because electron transport is inhibited by the cyanide. Cyanide gas classically has an almond smell, and I've put a picture of some almonds on the screen to remind you of that. And the anaerobic metabolism will cause lactic acidosis, which is one of the ways people can die from cyanide exposure. In modern clinical practice, one of the ways patients can potentially get cyanide poisoning is from nitroprusside. This is a hypertensive medication used to treat hypertensive emergencies. It contains 5 cyanide groups per molecule of nitroprusside, so if you expose people to this drug for long periods of time, they can develop toxic levels of cyanide. In actual clinical practice, we never give nitroprusside for more than a day or so because we don't want patients to get cyanide poisoning. And an interesting treatment of cyanide poisoning that ties back to electron transport is the use of nitrites. You can administer amyl nitrite to people who have been exposed to cyanide poisoning. So why does that work? Well, amyl nitrite converts Fe2+ to Fe3+ iron in hemoglobin. In other words, it creates methemoglobin, which is hemoglobin in the Fe3+ state. This will bind to the cyanide and protect the mitochondria so that they can start doing oxidative phosphorylation again, and this will help the patient get through the period of time until the cyanide washes out of their system. Now let's talk about uncoupling agents, and the way I think of these is that they essentially poke a hole in the inner membrane so that all those protons leak back out and they can't be used by ATP synthase to generate ATP. 2,4-dinitrophenol, or DNP, is used in research labs to uncouple oxidative phosphorylation. You can also uncouple oxidative phosphorylation if you take an overdose of aspirin. Patients who overdose on aspirin often have a fever, and the reason they have a fever is because you have uncoupled oxidative phosphorylation and all those hydrogen ions that are leaking out are generating heat. Another place where oxidative phosphorylation is uncoupled is in brown fat. This is fat that is found in newborns, but it goes away as the child grows up. It's also found in hibernating animals to keep them warm during the winter. Brown fat contains a substance called uncoupling protein 1. It's also called UCP1 or thermogenin. And sympathetic stimulation through norepinephrine and beta receptors leads to lipolysis of brown fat. Now, normally lipolysis would generate lots of ATP, but in brown fat, the electron transport is not coupled to oxidative phosphorylation. As a result, all those hydrogen ions leak out and they generate heat instead of ATP. This is very useful for hibernating animals in the winter. And as I said before, this is present in a small degree in newborns, but will resolve as the child grows up. And I summarized at the bottom of the screen here an important fact that all of these uncoupling agents lead to the production of heat because that energy in the form of protons is being used to generate heat as it leaks out instead of generating ATP. And then the last poison of oxidative phosphorylation I'll mention is a substance called oligomycin A. This is a macrolide antibiotic that is not used clinically. It inhibits ATP synthase, so protons cannot move through the enzyme. They are trapped in the intermembrane space. As a result, oxidative phosphorylation stops and ATP cannot be generated. And that concludes our module on electron transport.